The title of our manuscript is Mitochondrial Damps-Dependent Inflammasome Activation During Aging Induces Vascular Smooth Muscle Cell Dysfunction and Aortic Stiffness in Low Aerobic Capacity Rats. Most of the population studies on exercise capacity focus on correlation-based assessment of patients without attempting to infer a causal relationship between the variables. Experimental validation of causal inference, if any, in correlation-based population studies was performed using unique rat models with high and low intrinsic aerobic capacities. Specifically, this was accomplished by genetic segregation via two-way artificial selection based on treadmill exercise tests to investigate the influence of intrinsic aerobic capacity on all-cause mortality and morbidity. In addition to the 28 to 45% shorter lifespan, the LCR rats also exhibit a number of cardiovascular risk factors and impaired mitochondrial oxidative pathways compared to HCR rats. Since low aerobic capacity and aging have been linked to mitochondrial oxidative stress and dysfunction, we investigated whether low aerobic capacity runners have vascular dysfunction compared to high capacity runners. Our main observations include the following: altered transcription of genes of pro-inflammatory pathways, impaired mitophagy and oxidative phosphorylation pathways in old LCR aortas relative to age-matched HCR aortas. Secondly, we found that impaired arterial relaxation and increased aortic stiffness in old LCR rats compared to the HCR rats. Thirdly, increased mitochondrial oxidative stress, DNA damage, and decreased mitophagy in the aortas of aged LCR versus HCR rats. Further, increased expression of AIM2 inflammasome and its downstream effector caspase-1 in the aortic medial layer, as well as in VSMCs of old age LCR rats. Mitochondrial DNA damps treated VSMCs from young LCR rats showed similar expression and activation of AIM2 inflammasome relative to old LCR rat VSMCs. Rapamycin and MitoTEMPO attenuate inflammasome activation, mitochondrial oxidative stress, and improve oxygen consumption rate and mitophagy in old age LCR rat VSMCs. Our findings suggest that impaired mitophagy and inflammasome activation in the vasculature under conditions of low aerobic capacity, low aerobic exercise capacity during aging results in arterial dysfunction and aortic stiffness. The findings of our study indicate that in older adults with reduced aerobic capacity, mitochondrial antioxidants or mitophagy induction and or inflammasome inhibition may be effective therapeutic strategies for enhancing vascular health.